This is the beginning of the greatest battle of our century, the battle against complex diseases at the level of our own cells. Can we learn how diseases evolve? Can we predict complex diseases before they emerge? Can we reprogram our own disease fate? This is the story of humanity's struggle against the most profound and difficult challenge in our own history. Imagine that diseases are like movie, and maybe we have the early part of the movie, and then this healthy state actually go through intermediates. Then we have the disease condition, and the question is, what exactly happened between the healthy condition and disease condition? And can we build reasoning agents which enable us to predict and reconstruct these hidden or unseen intermediate conditions? So this is basically the question. Today I will be presenting a novel architecture of how we could build reasoning agents on biological manifold. And I believe this would be a unique path toward predictive and reprogrammable medicine. So I have been developing the disease state trajectory model, which state that over our lifetime, we start to be in healthy cellular states and we start to accumulate genetic and epigenetic dysregulations, which lead to manifestation of a dysregulated cellular state, which has high chance or high probability to branch to full-blown disease-specific trajectories. And diseases could actually take maybe 10 or 20 years to evolve, and what we do have currently from clinic or publicly available or maybe clinical cohorts, we have a snapshot of the healthy states as well as the disease condition, but the intermediate states are actually missing from our data. And these intermediates are super crucial because they can actually help us to predict future cellular events, and maybe we can predict the occurrence of cancer or other complex diseases maybe 5 or 10 years before the disease emerge, as well as we could use such cellular states to maybe freeze them to some interesting clinical outcome, or maybe we can reverse them back to be healthy-like conditions. I believe that in our field, in the medical domain as well as biological research, what we are trying to achieve is to define causal mechanisms of underlying biological processes and systems. And what I'm trying to argue is that maybe we have multiple causal paths which lead to the same clinical outcome. So you might have maybe lung cancer patients, they might have the same phenotypic outcome, but the underlying molecular events and the causal path led to the disease condition is quite unique and distinct from one patient to another. And I believe we need to build new models which is able to dissect and define these patient-specific causal paths so we can actually predict and intervene early on. So as I mentioned, you could have or you could build a healthy atlas cellular state describing the cellular state in a healthy condition as well as in the disease state, but the underlying continuous process is currently missing. I have been intrigued by major 3 questions of first, how we could reconstruct intermediate cellular state in the disease context from a snapshot single cell genomics data. How could we learn causal structures and gene regulatory network underlying cell types and states over long-term biological processes which might take months or years? And can we start asking counterfactual questions or what if questions for observational single cell genomics data use? So you could imagine that maybe we could start asking, what if I perturb this gene or this combination of gene? What might be the phenotypic outcome at the cell level and the disease level? So I started to formulate this problem as follows. So you could imagine that we have a true evolving biological process over time, and we do have a different time point describing this evolving process. And what we could do is that we build a predictive model where we could show time point 1 and time point 4, and we ask our agent to reconstruct the intermediates. And now we have an interesting observation where we could have such a reconstructed state and compare it back to time point 2 and time point 3. And if our agent or our model predict or reconstruct the right cellular states, we will have some sort of estimate and way of estimating if such reconstructed state are biologically meaningful by actually comparing them to time point 2 and time point 3. And if we have such type of models and agents, we'll be able to have a glimpse about these missing continuous processes. So for this, I developed Cell-DRL. This is a conceptual and algorithmic framework of a deep reinforcement learning agent that is capable of geometric reasoning on top of single-cell genomics data. So maybe we can have a bit of like introduction for reinforcement learning. So simply imagine that we have an agent is like a child, and this child to try to figure out the underlying dynamics of a certain environment through taking random actions. And after each of these actions, our agent start to collect observations, and given such observations, we give our agent either positive or negative rewards. So if our agent do the right thing given the objective function, we give our agent positive reward, and if the agent did the bad things, we give our agent a negative reward, and so on. So again, imagine that we have a huge manifold of health and disease related cellular states, and on top of this manifold we do have real data reflecting actual single cell genomics data, or maybe some clinical data representing the healthy intermediate condition as well as the disease condition. And as you can see here, we have like these black dots representing the real data. And the task of our agent is to connect these dots and basically try to generate these intermediate paths which connect the different cellular states on top of this manifold. And here I will just like zoom in into a two cellular state on top of this manifold. And basically here's our environment and cell-DRL environment architecture, where we start with our agent here, and then our agent start to take random actions and generate novel cellular states. And basically we define our initial and target state, and defining this reconstruction area where if our agent generate novel cellular states which is closer to the target state, we give our agent positive reward. And if the agent generate novel cellular state which is far away from this reconstruction area, we give our agent a negative reward. And we do this process iteratively for hundreds and thousands of times till the agent figure out the right sequence of actions to reconstruct the underlying trajectory. So basically I developed the architecture and represented the architecture as a Markov decision process, where we can define the MDB here by tuple of state and action probability and some reward signal. And basically our agents start to collect observations, and we push such observations to our policy network. And after each of these, and our policy start to emit actions, and we push such actions to our generative model, where it generates completely novel cellular states. And we can define our trajectory as a sequence of state and actions in the environment. And the core idea here is to build a reward model where this model is dependent on the current state of the environment, the just taken action, and the next state of the environment. And the goal of our agent is to maximize the cumulative reward over trajectories. And we try to define and learn policies which will maximize the overall reward over all trajectories. We did actually many in-silico validations in 8 biological scenarios, some of them like the cell cycle, epithelial to mesenchymal transition, in hematopoiesis, in tumor evolution, in multiple myeloma mouse model, as well as human multiple myeloma, and finally in direct cellular reprogramming context from fibroblast to cardiomyocytes. For today, I will just focus on the hematopoiesis example, in the human example, and the multiple myeloma example, as well as the direct cellular reprogramming. So in the hematopoiesis, as you know, we start actually in our body and in our bone marrow, we start with having hematopoietic stem cells, and these cells are actually going through multipotent progenitors, and these cells start to develop into more mature progenitors like neutrophil progenitors, erythroid progenitors. And basically these cells then will mature further to more mature cell types like fully mature erythroid cells or neutrophil cells and so on. So what we challenge the agent with is basically try to hide completely these intermediates. So simply showing the agent cluster 1 and cluster 2, so basically this erythroid progenitor part, the neutrophil progenitor part, so the two distinct parts of the movie, and completely hiding these intermediates, and ask our agent to reconstruct these intermediates. This is through the learning or the model training part. So basically, Cell-DRL agent go through two major phases. So basically the exploration phase and exploitation phase. In the exploration phase, the agent generate like crazy some cellular state which is far away from the target state. And here, as you see, we have like high cellular state loss. And over maybe 400 steps, the agent start to go through this exploitation phase where the agent successfully generate novel cellular states, which is exactly in this reconstruction area. And after the training, you could ask your agent to generate completely novel cellular state and manifold. And as you can see here, this is your cluster 1 and this is cluster 2. And here is the generated data, reconstruct the entire process, basically cluster 1, 2, as well as the entire intermediate conditions. We did lots of evaluations for the generated cellular state, and as you can see at the low dimension space, you can see that we have really nice co-clustering between real and generated data, as you can see in cluster 0 as well as the other clusters. And at the high dimension space, at the gene expression level, you can see that we have really nice correlation coefficients and correlation patterns between the real and generated genes within each of these subclasses. And then I started to ask this question of how can we know if actually we are able to reconstruct key marker genes which we know that are super crucial for the intermediate condition. And as you can see here, we do have, for example, in the real data, expression of CD34 plus CD48 and other marker genes, and actually Cell-DRL is able to reconstruct exactly this combination of genes. Also, I start to ask if we can capture correctly the underlying developmental order of these biological processes, and simply as you may know, actually this is the pseudotime ordering of the generated data, and here the pseudotime start with ELAN expression, MBO, and other neutrophil related progenitors, and then later on you see that we start to have this CD34 expression plus other intermediate state related genes which start to kick in the intermediate time point, and then later on we have this exclusive expression pattern between GATA2 and GATA1. Together with GATA1 we have the expression of CAR1 and CAR2. Basically this means that the agent is able to reconstruct correctly the underlying temporal order of these biological events. So I'm shifting gear now to the tumor evolution part, where we have the data for individual patient over the disease evolution over 3 years. So basically we have the primary condition, so the early part of the disease movie, where we have full-blown disease condition. This is actually the green color clusters. And then we have few cells, this is the remission condition. And then after almost 2 years, the patient experienced relapse 1, basically this few distinct cluster here. and then we have after a few months we have relapse 2. And and basically you could actually see here that we have all this white space. So basically it means that we have missing cellular state as well, because this is 3-year process and we just have 4 different time points. So to challenge the agent, I start to show the agent the primary condition as well as relapse 2 and hide completely the intermediate data points, and train the agent and ask the agent to reconstruct the intermediates. And as you can see here, the agent was able to reconstruct and generate the intermediate cellular states, which actually they are not really present in the data. So they are co-clustering with the relapse 2, the remission 2, remission condition. So basically the intermediates, as well as generated completely novel cellular state, which is not present in the data. And if we go back to the generated gene expression matrix, you can find specific marker genes like SPARC1, CXL12, and other genes like NOPR1, NOTCH3, and all these genes are actually governing really crucial processes like proliferation, survival of the tumor cells, increasing osteoclastogenesis, angiogenesis, as well as bone microenvironment remodeling. And imagine if we have such information early on, maybe we'll be able to stop completely the experience of relapse 2 for such patients. And for this part, I will be presenting the experimental validation part to really show that these agents can translate directly to meaningful biological representations, and maybe we can use this technology to reprogram cell types and states for complex diseases. So here we started to ask this question of how could we navigate the cellular state landscape. So imagine that you would like actually to reprogram one cell type to another identity. One way is to go through this indirect reprogramming path. So basically try to reprogram this identity to its own intermediate progenitor, and you reverse it back to pluripotent stem cell. And then this pluripotent stem cell, you could reprogram it to the other identity intermediate progenitor, and then you could push it to the new identity. But basically this process, it means that it's an indirect process, and you need to reprogram most of these identities to go through this pluripotent stem cell state. And most of the time is super harsh actually to make this working. And some other ways is to maybe try to find some sort of direct reprogramming path between one identity and another. And basically the people doing this now by like randomly selecting a combination of genes which try to reprogram one identity to another. So is there a way maybe we can use our agent to rationally design and reprogram one cell state or one identity to another? So I tried to play with this idea in the myocardial infarction process, where we could have a healthy heart, and this heart could experience infarction event, and the patient would go through acute phase maybe for 6 hours, and then fibrogenesis phase, which would last for many weeks. In the acute phase, the patient would experience ischemic, maybe necrotic-like tissue and ischemic zones. And in the fibrogenesis phase, the patient would experience fibrotic zones, basically scar-like tissues. And naturally or normally, the cardiomyocytes will be lost, and the cells will be dying, and this would be replaced by cardiac fibroblast populations. And the question is, can we reprogram such fibroblast populations to be toward the cardiac fate? And the core idea here is to define a causal path which might govern the fibroblast-to-cardiomyocyte transdifferentiation. So with this, I tried to challenge the agent by showing the fibroblast identity and the cardiomyocyte identity, and ask the agent to predict maybe novel path, which, who knows, maybe it doesn't even exist in nature, and try to validate this experimentally to see if such paths could naturally exist if we tried experimentally. And this is how it looks like. So basically this is the fibroblast, this is our cardiomyocytes, and all this green color cells is actually the generated Cell-DRL generated transdifferentiation path, or the direct reprogramming path. And then I have been looking in depth into these transition states, and I was able to define mainly three major key transcription events which regulate the fibroblast reprogramming toward cardiac or cardiomyocyte fate. Basically, we have this first biological program which start to kick in the fibroblast identity toward the intermediate state and start to be downregulated in this intermediate state. We have a completely new biological program which is specific for the intermediate state, which start to kick in basically by the end of event 1 and start to be expressed up to the end of event 3. And then we have a third event basically pushing these cells to commit to the cardiomyocyte fate. And then I defined basically three major biological programs and genes governing this process, and mainly these intermediate genes. And I started to design experimental layout to try to sequentially induce these events experimentally together with my colleague Andy. So basically we start with the cardiomyocytes, and we add event 1 high L ligand, which is DDL4 protein, for 2 days, and then we add event 2 genes, basically the lentiviruses with our predicted transcript factors and DDL1 protein. And then over this process, after day 7, day 14, we start to do a qPCR and imaging as well as some other readouts. So after day 7, we start to observe actually downregulation of cardiac or fibroblast related markers like COL1A1, FN1. Basically, it means that these cells start to lose their own initial identity marker genes. And then we start to observe upregulation of cardiac and cardiac progenitor identity genes like NKX2-5, MYH6, and so on. And then we start to look at functional genes like potassium and calcium channel, and we are observing upregulation of key marker genes connected to potassium and calcium channel, meaning that these cells start to acquire functional units toward the cardiac fate. Also, we did a FAXA analysis, and we start to observe TNT expression as well as alpha-actinin signal in our event 2 condition. And then I tried to map the single cell data from our in vitro event 2 intermediate state at day 7, and they are co-clustering perfectly with the generated intermediate state, meaning that we are able actually to predict such novel reprogramming path, and we are able experimentally to show that they are there, and we can map them back to the intermediate generated condition. Also, we did bulk sequencing data for the intermediates, and again we observe downregulation of fibroblast related genes as well as upregulation of cardiac related markers. And at day 14, we start to do imaging, and we observe upregulation of cardiac markers like alpha-actinin, for example, as well as increase in the mitochondrial network branching, which is one of the characteristics for cardiac fate. I believe that we presented AI system which enable us to reason on top of biological manifolds, as well as a way of maybe reprogramming and reversing cellular state, as we showed experimentally in the cardiac fate. And I hope such technologies will enable us to maybe expand it to many complex diseases, as well as proposing a way to achieve predictive and reprogrammable medicine. Good question. So Cell-DRL is just like cell, DRL, deep reinforcement learning. That's it.